Something is happening inside your body right now, tonight, between 2 and 4 in the morning, not eventually, not if things go wrong, right now, on a schedule your body has run every night since you were born. During that 2-hour window, your pituitary gland releases the highest pulse of human growth hormone your body produces in a 24-hour period. Your stem cells, the repair workforce embedded in your bone marrow, your gut lining, your muscle, your brain, are supposed to be at peak activity during that window, dividing, differentiating, replacing damaged tissue, running the maintenance cycle that determines how fast or how slowly you age at the cellular level. Here's the problem. After 60, for the majority of people living with chronic stress, cortisol doesn't drop far enough at night to let that repair window open properly. The hormone that's supposed to be at its daily minimum between midnight and 4 a.m. is instead sitting at levels that suppress the exact biological processes your body needs to run its repair shift. And most people have no idea this is happening. They sleep, they wake up, they chalk up the fatigue, the slower recovery, the accumulating wear to getting older. But the mechanism is specific, it's measurable, and it's addressable. I'm Dr. Bennett. Today, I want to walk you through what I think is the most underappreciated driver of accelerated aging in people over 50. Not what you eat, not what you're exposed to in the environment, but a hormone your own body produces quietly in the wrong pattern while you sleep. And then I want to show you what the research says about a specific intervention studied in over 20 randomized control trials, with a 60-day cortisol reduction in one landmark study of 27.9%. Before I go further, I need to be clear. This video involves discussion of hormones, adrenal function, and cellular aging that overlaps with medical conditions, including Cushing syndrome, hypothyroidism, and autoimmune disease. Everything I'm sharing is educational, based on peer-reviewed research, and is not a diagnosis or treatment plan. If you're under medical care for any endocrine or immune condition, or taking immunosuppressants, sedatives, or thyroid medication, please bring this information to your physician before making any changes. For years, the conversation around cortisol in medicine has been dominated by two extremes: Cushing syndrome on one end, severely elevated cortisol from tumors or prolonged steroid use; Addison's disease on the other, dangerously low cortisol from adrenal insufficiency. The vast middle ground where most adults over 60 are actually living has been largely ignored. Cortisol that's chronically elevated, but not at disease levels. Disrupted in its daily rhythm, too high at night, too blunted in its morning peak. This zone doesn't show up as an abnormal result on standard blood panels. It doesn't produce dramatic symptoms. It produces the kind of changes we tend to accept as normal aging: persistent abdominal weight gain, fatigue that doesn't respond to sleep, memory that's slightly worse than it was 5 years ago, recovery from illness or injury that takes noticeably longer. What researchers now call functional hypercortisolemia has direct, measurable consequences for stem cell populations. And once you understand the mechanism, the urgency becomes clear. Your body contains multiple populations of stem cells. The most relevant here are your mesenchymal stem cells, MSCs, housed primarily in bone marrow. These are responsible for regenerating bone, cartilage, muscle, tendon, and connective tissue. They are the structural repair crew. In a healthy 25-year-old, MSC populations are abundant and highly responsive. By age 60, those populations have already declined to roughly 30 to 40% of their peak levels. Cortisol speaks directly to these cells through glucocorticoid receptors embedded in their membranes. When cortisol binds to them, appropriate in short bursts during actual stress, the MSC essentially receives a signal to stand down. Don't divide, don't differentiate. Conservation mode. This is a perfectly functional short-term mechanism. The problem arises when the signal doesn't turn off. When cortisol is chronically elevated day after day, week after week, year after year, MSCs receive a continuous stand-down signal. The pool of available stem cells for tissue maintenance shrinks. And in someone already at 30 to 40% of their youthful MSC reserve, that additional suppression is the difference between a body that can still repair itself adequately and one that cannot. The molecular mechanism goes deeper. Cortisol binding to glucocorticoid receptors suppresses the transcription of pluripotency factors, the genes that determine a stem cell's ability to differentiate into multiple cell types. The key factors are OCT4 and NANOG. When their expression is suppressed by chronic glucocorticoid signaling, stem cells lose flexibility. They become less capable of responding to the body's repair signals, and more likely to shift toward pathways that produce fat cells rather than bone or muscle, which helps explain the characteristic body composition changes in people with elevated cortisol over time. There's a second mechanism that operates on a longer timescale with even more permanent consequences. Chronic stress and elevated cortisol accelerate telomere shortening. Telomeres are the protective caps on the ends of chromosomes. They shorten with every cell division, and when they become critically short, the cell either enters senescence and stops dividing, or undergoes apoptosis and dies. A meta-analysis published in the Proceedings of the National Academy of Sciences found that people with chronic psychological stress had telomere lengths corresponding to 9 to 17 additional years of biological aging compared to low-stress controls. That's the difference between cells behaving like they belong in a 50-year-old body versus a 67-year-old body in the same person, depending on their cortisol history. Cortisol also degrades gut barrier integrity. The intestinal crypt stem cells, the fastest cycling stem cells in adult tissue, renew the entire gut lining every 4 to 5 days. Chronic cortisol elevation disrupts the tight junction proteins that seal gaps between epithelial cells. As those junctions loosen, bacterial compounds cross into systemic circulation, triggering immune activation and feeding back into systemic inflammation. This explains why people with chronically elevated cortisol often develop bloating, irregular bowel habits, and food sensitivities in middle age with no obvious dietary explanation. The problem isn't in the diet. It's in the hormonal environment degrading the gut's repair capacity. In healthy adults, cortisol follows a precise daily rhythm. It peaks about 30 minutes after waking, the cortisol awakening response, then declines gradually and reaches its minimum between midnight and 2 a.m. That minimum is critical because the 2 to 4 a.m. window is when growth hormone secretion peaks. Growth hormone tells MSCs to divide, tells muscle satellite cells to initiate repair, tells neural stem cells in the hippocampus to produce new neurons, a process called neurogenesis that supports memory consolidation and cognitive resilience. High cortisol at night doesn't just disrupt sleep quality, it directly competes with the growth hormone signal. When cortisol is elevated during the window that should belong to growth hormone, the repair cycle is muted. The night crew shows up, but the work orders have been canceled. Restoring the normal nocturnal cortisol minimum doesn't double your stem cells. It restores the nighttime window in which your existing stem cells do the work they are designed to do, work that has been systematically suppressed by cortisol that isn't dropping the way it should. There are 5 patterns that, in people over 50, are highly suggestive of disrupted cortisol rhythm. None are diagnostic on their own. A physician evaluation, and ideally a diurnal salivary cortisol test, is needed for that. But if 3 or more describe your current experience, this conversation is directly relevant to your biology. The first is waking between 2 and 4 in the morning without a clear reason. The cortisol surge that should happen gradually before dawn is happening too early and too aggressively, interrupting sleep in what should be the most restorative hours. The second is persistent abdominal fat that doesn't respond proportionally to diet or exercise. Cortisol drives fat deposition specifically in visceral adipose tissue through mechanisms involving direct glucocorticoid signaling in adipocytes. The third is memory declining faster than expected, specifically working memory and the ability to retain new information. Chronically elevated cortisol shrinks hippocampal volume and suppresses the neurogenesis that maintains hippocampal function. The fourth is the tired but wired feeling, exhausted enough to want sleep but unable to actually fall or stay asleep. The fifth is getting sick more often, or taking significantly longer to recover. Chronic cortisol suppression of hematopoietic stem cell function accelerates immunosenescence, the age-related decline in immune function that makes older adults more vulnerable to infection. The research on ashwagandha, specifically the standardized root extract known as KSM-66, represents one of the most robust bodies of evidence for any adaptogenic compound in the medical literature. A 2025 systematic review and meta-analysis published in a peer-reviewed journal analyzed 7 RCTs on cortisol specifically, involving 488 participants, conducted through a rigorous PRISMA-compliant search across PubMed, Web of Science, Scopus, and the Cochrane database. The conclusion was a statistically significant reduction in serum cortisol with ashwagandha compared to placebo. The landmark individual trial, The Chandrasekhar study, published in the Indian Journal of Psychological Medicine, was a prospective, randomized, double-blind, placebo-controlled design involving 64 adults with chronic stress over 8 weeks. The dose was 300 mg of KSM-66 twice daily, totaling 600 mg per day. The result on cortisol, a 27.9% reduction versus placebo. That is not a marginal effect. Pharmaceutical interventions for stress-related cortisol elevation are relatively limited and carry significant side effect profiles. A 27.9% reduction from a well-tolerated botanical extract in a properly controlled trial is a clinically meaningful finding. Sleep quality improves significantly as well, closing the loop on the nocturnal cortisol mechanism. Lower cortisol during sleep restores the growth hormone window. The repair shift runs. A second study adds a dimension particularly relevant for men between 40 and 70. This double-blind, placebo-controlled crossover trial showed an 18% increase in DHEAS and a 14.7% increase in testosterone versus placebo, both statistically significant. DHEA, produced by the adrenal glands, functions as a physiological antagonist to cortisol. When cortisol is chronically elevated, DHEAS tends to decline as the adrenal glands prioritize cortisol production. The cortisol to DHEA ratio is one of the most reliable biological indicators of chronic stress burden and biological aging rate. Restoring DHEAS while simultaneously reducing cortisol shifts this ratio in the favorable direction, and the effects on stem cell function are direct. A 2025 review in Biogerontology synthesized ashwagandha's aging protective mechanisms at the cellular level. Withanol, one of the active withanolides, activates heat shock proteins, molecular chaperones that prevent protein misfolding inside cells. Protein misfolding is a central feature of cellular aging and directly relevant to stem cell dysfunction. The same review documented CRP reductions of over 30% alongside reductions in IL-6 and TNF-alpha. Ashwagandha is not acting through a single narrow pathway. It is working on several intersecting loops simultaneously: cortisol, inflammation, and cellular protein quality control. The review also described evidence for improved mitochondrial function and neurogenesis promotion through BDNF upregulation, supporting the same hippocampal neural stem cells that chronic cortisol suppresses. A 2025 safety study in phytotherapy research followed 191 adults taking standardized ashwagandha for 12 consecutive months, monitoring liver function, kidney function, thyroid markers, and hematological parameters. The conclusion: no clinically significant changes in any of those parameters. An adverse event rate of 9.4%, comparable to placebo in similar studies. 12 months of daily use, nearly 200 subjects, comprehensive monitoring. This is the evidence profile that distinguishes a supplement worth considering seriously from the vast majority of compounds on the market. That said, ashwagandha is contraindicated in pregnancy. If you have an active autoimmune condition, rheumatoid arthritis, lupus, Hashimoto's, multiple sclerosis, discuss it with your physician first, as its immune-modulating properties can theoretically exacerbate autoimmune activity. If you take thyroid medication, ashwagandha has been shown to increase serum T4 levels, which can push someone outside their therapeutic range. And if you take sedative medications, benzodiazepines, sleep aids, certain antihistamines, the additive CNS depressant effects require medical oversight. The protocol I would walk a patient through focuses on restoring normal nocturnal cortisol rhythm and giving stem cells the conditions they need to do their repair work. The foundation is ashwagandha KSM-66 at 600 mg daily, either split as 300 mg with breakfast and 300 mg in the evening, or taken as a full 600 mg approximately 1 hour before sleep. Look specifically for KSM-66 or Sensoril on the label. These are standardized extracts validated in the clinical trials. Generic ashwagandha products using whole herb powder do not have consistent withanolide content. Expect 4 to 6 weeks before the cortisol effect is substantial. Withanolides don't block cortisol production the way a pharmaceutical would. They restore the responsiveness of feedback regulation that tells the adrenal glands when to stand down. That's a more physiological, more sustainable effect, but it's not immediate. The second element is magnesium glycinate at 300 to 400 mg taken approximately 1 hour before sleep. Magnesium is a cofactor for over 300 enzymatic reactions, including many that regulate the transition from sympathetic to parasympathetic dominance, from the alert, high cortisol state to rest and repair. The glycinate form has superior absorption in older adults. Beyond autonomic regulation, glycine is a direct precursor to glutathione, the master intracellular antioxidant, meaning magnesium glycinate supports stem cell DNA integrity as well. If you're on diuretics or have kidney disease, have your physician check magnesium levels before adding supplemental magnesium. The third element is phosphatidylserine at 100 to 300 mg daily. This phospholipid concentrated in neuronal cell membranes blunts the cortisol response to acute stressors without eliminating cortisol's normal function. Multiple RCTs in adults over 50 have shown it reduces the peak cortisol spike in response to exercise and psychological stress. Beyond supplements, the behavioral interventions are equally important. Diaphragmatic breathing in the evening, specifically the 4-7-8 pattern, inhaling for 4 counts, holding for 7, exhaling for 8, activates the vagus nerve and directly reduces HPA axis activity. 5 cycles before sleep takes less than 3 minutes and has documented effects on heart rate variability that translate to measurable reductions in nocturnal cortisol. Sleeping in a room between 18 and 20 degrees Celsius supports the thermoregulatory drop that triggers growth hormone release. And 1 hour of blue light exposure in the evening has been shown to elevate that night's cortisol by an average of 37%, meaning every additional hour of screen time after dark is actively maintaining a cortisol environment incompatible with the repair window you're trying to restore. On exercise, high-intensity intervals and heavy resistance training produce a significant cortisol spike. In adults over 60 with already compromised HPA regulation, that spike can persist and extend into the nocturnal period. Moderate intensity activity, walking 30 to 45 minutes, swimming, yoga, tai chi, reduces basal cortisol over time without triggering disruptive spikes. The goal isn't to avoid exercise, it's to choose the type and timing that works with your cortisol biology rather than against it. For monitoring, the most informative test is a diurnal salivary cortisol profile, saliva samples at wake, midday, late afternoon, and bedtime. A single morning blood draw tells you almost nothing about rhythm. Normal evening values should be below 100 nanomoles per liter. Above 200 at night is functional hypercortisolemia, not a disease diagnosis, but a biological state actively impairing your repair cycle. On a realistic timeline, sleep improvements often begin within the first week as magnesium glycinate and behavioral changes take effect. At 4 to 6 weeks, the ashwagandha effect on HPA axis sensitivity becomes substantive. People describe less reactivity and faster recovery from stress. At 2 to 3 months, the changes that matter most for stem cell function are underway. Reduced cortisol burden, improved DHEAS ratio, lower inflammatory markers, shifting the bone marrow environment in the right direction. The thread running through everything we've discussed is the biological environment for your stem cells, all of them, MSCs, hematopoietic stem cells, intestinal crypt stem cells, hippocampal neural stem cells, are sensitive to the chemical signals surrounding them. Cortisol is among the most potent of those signals, and chronic elevation of it is among the most tractable modifiable factors driving premature stem cell decline. Every night that passes with your cortisol in a healthy rhythm is a night the repair shift runs properly. After 60, that is not something to leave to chance. Leave a comment below with your biggest question from today, or let me know which symptom pattern resonated most. I read everything, and it directly shapes what I cover next. Keep asking the right questions. I'll see you in the next one.